What I wanted to share today is kind of my lab's approach to next generation RNA vaccines and therapies. So kind of where we are in the field and where we're going. So just to give you an outline of what I'm going to talk about today, I'll talk about the development of saRNA vaccines and give a brief overview of self-amplifying RNA for those of you who aren't familiar. I'll talk about the effect of formulation on the expression and immunogenicity of self-amplifying RNA. I'll talk about some work we've done in my lab to optimize lipid nanoparticle delivery of both mRNA and saRNA, and then actually deviate probably from the nanomedicine approach and talk about some RNA design and how we can use that to affect the intracellular sensing of these nanomedicines. So just to declare my conflicts of interest, I'm co-founder of a company called VaxEquity, which develops saRNA vaccines. So keep in mind that I'm very biased. I'm also on the scientific advisory board for GenVax, and I consult for two companies, Replicate Biosciences and the Cystic Fibrosis Foundation. So it's been a really exciting time to be in the field. Whenever I used to start giving these lectures prior to the pandemic, I would always have to spend about the first half of the lecture just telling people what mRNA vaccines were and trying to convince them that they would work one day. So obviously a lot has changed in the past 3 or so years with the approval of 2 mRNA vaccines. So I like to say, the plane is off the ground, but where do we go with this technology from here? I think it's a really exciting challenge. So in my lab at UBC, we have really kind of 3 different focuses. So the first is RNA molecular engineering. So how do we design the RNA to express for longer or express in certain types of cells? How do we design better nanoparticles so we get this RNA to the cells and tissues where it needs to go? And then thinking about the next frontier of RNA and designing better vaccines and using RNA technology for other applications. So some of the things we're working on now is making other vaccines, for example, for a bacterial pathogen instead of a viral pathogen, gaining a better understanding of the immune response to RNA vaccines, and using RNA to express monoclonal antibodies, which can be used to treat cancer or prevent infectious diseases. So one of the things I wanted to start off talking about was really the priority areas in the field. So one of the main things that we're trying to do right now is really minimize the dose of RNA to reduce side effects. So I wanted to share with you some of Moderna's clinical trial data where they were looking at a phase 1 trial. So the primary outcome of this kind of trial is the safety. And they were looking at 3 different doses of their mRNA vaccine. So you can see here 25, 100, and 250 micrograms. They categorized the side effects that they observed in study participants as mild in gray, moderate in blue, and severe in yellow. And I think what's very visibly apparent from this data is that as you increase the dose of RNA, you also increase both the frequency and the severity of side effects. So there's lots and lots of clinical trial data out there on mRNA vaccines now, and this is a very clear trend in all of this data. So what this really means to us is that we really need to think about strategies to minimize the dose of RNA that's required so that we can continue to use mRNA in the context of non-pandemic vaccines, such as an annual influenza vaccine, as well as other applications of RNA, such as RNA therapeutics. Oops. So one of the ways we can do this is with a technology called self-amplifying RNA. There's lots and lots of different approaches to minimize the dose of RNA, but this is one that my lab focuses on. So to walk you through the structure here and kind of the main differences, so we have at the top a conventional messenger RNA. So at the 5' end, we have a cap, and at the 3' end, we have a poly A tail. We also have an untranslated region at both the 5' and 3' ends, and then our antigen code here in the middle. Self-amplifying RNA has a lot of similar structural components. You'll notice it has the same 5' cap and 3' poly A tail, as well as the 5' and 3' UTR and the antigen code. The main difference is in the middle, these 4 different proteins that are also expressed, or NSPs, or non-structural proteins. So these are derived from an alpha virus and encode a replicase enzyme. So what this means is that once that RNA enters a cell, it's able to copy itself. So I have a very simple schematic here at the bottom. So on the right, we have messenger RNA. So once an mRNA enters a cell, it engages with the ribosome and you get translation of the antigen. saRNA starts out very similarly. Once it enters a cell, it engages with the ribosome. From this engagement, you do get some translation of the antigen, but more importantly, you get translation of this replicase enzyme. So this replicase enzyme then goes back up and engages with this original strand of saRNA and makes exact copies of it. So I have in the schematic just 3 copies depicted here, but it actually produces thousands of copies of the RNA. So because of that, you get more translation of your antigen. So this means that we can typically use a much lower dose of saRNA than mRNA. It's about 100 times lower. One of the other differences to keep in mind is that saRNA is much longer than mRNA, so it's about 10 times as long. If a typical mRNA is around 1,000 nucleotides, a typical saRNA is about 10,000 nucleotides. Of course, one of the biggest challenges that we're probably all aware of is getting these large negatively charged molecules into a cell, which doesn't happen spontaneously. So we really have to design systems to get them taken up and then expressed in cells. One thing I wanted to show that many people may not be aware of as well is actually saRNA also results in a lot longer duration of protein expression in vivo. So this was a study that we did in mice using a firefly luciferase as a reporter protein. And we made an mRNA and an saRNA formulated in the exact same lipid nanoparticle formulation, and then injected at a dose of 1 microgram. So on the y-axis, we have the total luminescence. So this correlates with the amount of protein expression that we see. And then we image the mice over the course of a month. And what's very clearly apparent from this data is that with the mRNA, you see really high expression really quickly, but this turns off around day 4 in this experiment. Whereas with the saRNA, it takes a little more time to get going to really kick off that replication and start expressing protein. And we see it peak around day 10, but we continue to see protein expression out to day 30 with a really kind of nice even fall off there. So one of the really interesting things about this is they just have very different protein expression profiles. So the saRNA is a little bit more delayed compared to the mRNA, but we get a much longer duration of protein expression. So I want to talk about kind of my experience with working on an saRNA vaccine and really highlight why this technology is so advantageous and kind of why it's such a great tool to have going forward for any future outbreaks or pandemics. So I'm sure everybody is very well familiar with this now, but I just wanted to talk through kind of the vaccine that I worked on as a postdoc at Imperial College. So how do you make an RNA vaccine against a brand new pathogen? So for those of you who aren't familiar, what we do is we take the spike protein, which is on the surface of the virus, SARS-CoV-2 in this case. We encode that in our saRNA. We then package that into lipid nanoparticles and then inject that into mice. And for this study, we were working with CQAS Therapeutics, which is right here in Vancouver, to work on the lipid nanoparticle formulations. So I think one of the most amazing things about RNA vaccines or RNA technology in general is just how modular they are and how quickly we can develop new vaccines. This doesn't mean there's any difference in the time it takes to test them, but it's actually quite easy to make new ones. So I wanted to walk you through our team's timeline of how long it took us to develop an RNA vaccine in early 2020. So the circulating strain of the wild type SARS-CoV-2 virus was sequenced and released to the public on the 10th of January, 2020. Our team accessed the sequence on the 20th of January. Very coincidentally, I flew to MSL, my interview here at UBC, on the 21st of January. So you can imagine this was an extremely busy time. We ordered the DNA oligos to make our RNA vaccine on the 22nd of January. We assembled the genes on the 28th of January. We then confirmed the expression of the saRNA vaccine on the 12th of February. And then on the next day, we started our immunogenicity studies in mice. We then started our rat toxicology study on the 30th of March. Because this was a first-in-man trial, we had to do this prior to starting our human clinical trial. So we submitted our paper on the 22nd of April, and then started our clinical trials on the 19th of June. Excuse me. So even as a small academic team, we went from designing a new saRNA vaccine to testing it in humans within 6 months. And people have done this even reportedly faster. So the team at Moderna did this in just about 66 days. So this is very different from how long it's taken in the past to develop new types of vaccines, which I think is really why the mRNA vaccines made such a difference during the pandemic. So I wanted to walk you through a little bit of data from our vaccine. So there's a lot of different groups here, but I'll walk you through them. So at the time we were doing these studies, there was no state-of-the-art mRNA vaccine because nothing had been approved before. So in the lab, we used electroporated DNA as our positive control. We used a different vaccine against rabies as our negative control. And then we used 4 different doses of our saRNA formulated in lipid nanoparticles at doses of 10, 1, 0.1, and 0.01 micrograms. And then we compared this to the antibody responses that we saw in recovered COVID-19 patients. So these mice got injections at 0 and 4 weeks, and then we measured the SARS-CoV-2 specific IgG using an ELISA at 2, 4, and 6 weeks. So you can see with our DNA positive control group that we get a good immune response, about 10 to the 5th nanograms per mil. And with our saRNA vaccine at the same dose of 10 micrograms, we get an even higher antibody response. So just over 10 to the 6 nanograms per mil with our highest dose, which goes down nicely in the dose response curve. So even at our lowest dose of 0.01 micrograms, we're seeing an antibody response of 10 to the 5th nanograms per mil, which was over an order of magnitude higher than the antibody responses we were seeing in recovered COVID-19 patients at that time anyways. So at that time, it was about 100 times higher antibody response than a natural infection. Of course, we have a lot more data on these antibody responses and the duration now. One of the things we also wanted to look at was, okay, so we're producing lots of antibodies. So are these functional antibodies? And so in order to do this, we ran a pseudovirus neutralization assay. So on the y-axis, we have the IC50. We also have this exact same groups here. So you can see with our DNA group, we see a pretty good neutralization, about 10 to the 3rd. And again, the response that we see with our saRNA vaccine is about 2 orders of magnitude higher. So about 10 to the 5th, again, a very nice dose response curve. And even this lowest group has a higher neutralization than what we were seeing in the sera from recovered patients. So one thing we were really interested to do was look at kind of this correlation between the titer of antibodies and the neutralization. So this is kind of like the quality versus the quantity of the antibodies. And we did this for both the mice and the patient samples that we had at the time. And you can see that there's a positive correlation between each. So as we're producing more antibodies, these antibodies are able to neutralize the virus, which is exactly what you want to see for a vaccine. One of the things that's also really interesting and I think kind of underappreciated about RNA vaccines, or you hear about them much less, is the cellular response that we see in response to RNA vaccines. So this is actually really profound and I think an important part of the immunity that still needs to be looked into more, kind of just beyond the humoral immunity. So in order to test this, we did an ELISpot assay using interferon gamma. So on the y-axis, this is expressed as the spot forming units per million splenocytes. So we take out the splenocytes, we re-stimulate them with the spike peptides, and then we look at how many of those splenocytes are secreting interferon gamma. So we have this at the bottom for our electroporated DNA, which is at about 250 spot forming units. And this may seem like a low number on this graph, but this is actually like a pretty good cellular response in mice. It's just totally eclipsed by the response that we see from our saRNA vaccine. So the cellular response from our RNA vaccine was about 10 times higher than this, so 2,500 spot forming units per million splenocytes. Again, we see a good dose response curve, and even at our lowest dose of 0.01 micrograms, we're seeing about 1,500. So in addition to producing a lot of antibodies, it also produces a robust T cell response, which is actually really important in the context of SARS-CoV-2 as well. So everybody always asks me, where is the ICL saRNA vaccine now? So we finished our phase 1/2 clinical trial in January 2021, right as I was starting here at Imperial or at UBC actually. And so at that point, the government, this is like basically all funded by the UK government. And at that point, they didn't want to fund these £130 million phase 3 trial. And so the team pivoted to targeting COVID mutation and booster doses. And we actually did a lot of work with Oxford looking at heterogeneous vaccination regimens with viral vectors as well. And then started a company to continue to develop this more, which now has lab space in Cambridge in the UK. And last year, I guess 2021, partnered with AstraZeneca to really develop this platform even more. So it's really exciting to see this technology really getting embraced by big pharma and really getting off the ground. And probably what's even more exciting is that there's actually an approved saRNA vaccine now, which was developed by HDT Bio in Seattle. So this is an saRNA vaccine against COVID-19, which was approved in India at a dose of 5 micrograms. So it's really cool to see this taking off and really starting to get more and more developed, which is great. So where do we go from here? Today, I'm going to talk about kind of two different aspects of our work. So both the nanoparticle and the RNA design side. And first, I wanted to start off thinking about the RNA delivery vehicles. So many people will be familiar when we're formulating RNA, we take a fluid of the saRNA, which is negatively charged, and we complex it with some sort of cationic or positively charged delivery vehicle. So when we're formulating this, this is most commonly done through a microfluidic system wherein we produce a nanoparticle with the RNA on the inside. So for saRNA specifically, there's really been kind of three different types of nanoparticles that have been used to deliver saRNA. So polymeric nanoparticles, lipid nanoparticles, which we're all familiar with, those are what's in the mRNA COVID-19 vaccines, and a nanoemulsion. And actually, the approved saRNA vaccine uses a nanoemulsion. So one thing that I've always thought about, and I'll come back to this a few times during this presentation, is the schematic that was published by Norbert Hardy in 2018. And what it's really looking at here is how are these nanoparticles or mRNA vaccines sensed intracellularly? And one thing that's always really stuck out to me is, in this kind of the main point is comparing these unmodified and modified mRNAs. And we know that the unmodified mRNAs are sensed by some endosomal sensing mechanisms like TLR3, 7, and 8, and cytosolic sensing mechanisms like MDA5 and RIG-I, which results in this type 1 interferon response. But what's really always stuck out to me, and I think we're still learning a lot in this field, is you'll notice over here there's a nanoparticle and it just says carrier sensing with a question mark next to it. So we now are starting to appreciate that the lipid nanoparticles actually have an adjuvant effect. We just don't really understand how the delivery vehicle is sensed by cells. So this is something that we were really interested in looking into. So our goal in this study was really to do a head-to-head comparison of polymeric and lipid nanoparticle formulations of saRNA. And I think one thing I've noticed in the field is people are typically, a polymer person or an LNP person. And so you don't actually see many of these head-to-head studies. But we were really interested in using kind of a state-of-the-art polymer and a state-of-the-art LNP to really get a good sense of how these compare for protein expression and the immunogenicity that we see with vaccines. So on the left, our polymeric system is called pABOL, which is a bioreducible cationic polymer that was developed in collaboration with Molly Stevens' lab at Imperial. And you'll notice these particles are quite simple. So it's just two components, the saRNA and pABOL are polymer. On the right, we have a typical lipid nanoparticle formulation, and this work was done in collaboration with Precision Nanosystems, also here in Vancouver, and which have many more components, which many of us will be familiar with. So we have the ionizable lipid, which actually complexes the saRNA, cholesterol, a phospholipid, and a pegylated lipid. And in this study, we use two different phospholipids, so DOPE and DSPC. So we'll annotate that throughout using PE or PC. So the first thing we wanted to do is really characterize the particle characteristics of these formulations. So I'll walk you through what all these groups are. So we have pABOL at the top, which is our polymer, and then our 5 different lipid nanoparticle formulations. So LM just stands for lipid mix 1, 2, 3, 4, and then formulated either with DOPE or DSPC. And what you can see here, we looked at the size, the surface charge, and the encapsulation efficiency. As far as the size goes, they're all around 80 nanometers with a polydispersity index of about 0.1. So very similar characteristics in that right. The main difference that we saw was the difference in the surface charge or the zeta potential. So you can see with our pABOL polymer that it's still positively charged, about +8 millivolts, whereas all of the lipid nanoparticles are neutrally charged. When we looked at the encapsulation efficiency, you can see that our pABOL is about 100%. One of the nice things about this formulation is it doesn't require any downstream purification after formulating. So we always get 100% encapsulation, whereas the lipid nanoparticles were all about 84% for the saRNA in this case. So the first thing we wanted to do was look at how much protein expression do we see after an intramuscular injection of all of these formulations. So again, we have our pABOL polyplexes and then our 5 different lipid nanoparticle formulations. This was using an saRNA encoding firefly luciferase. So again, on the y-axis, we have the total flux or luminescence. And what you can see here is we've done the statistics, but I think this data really speaks for itself. So with the pABOL, we see just over 10 to the 6th for the flux, whereas for the lipid nanoparticles, we see about 10 to the 4th for the expression. So we see about 100 times higher protein expression intramuscularly with our pABOL formulation. And one trend that we saw throughout is that actually for these formulations, the DOPE seemed to express better than any of the formulations with the DSPC. So we then went on to look at whether or how these two delivery systems compare as a vaccine. And so one of the things we wanted to test was, okay, so using influenza hemagglutinin as a model antigen, how do they compare when we're looking at the antibody titers and then in a challenge study? So on the left is the antibody titers from this study. So again, we have our pABOL polyplexes and our 5 different lipid nanoparticles. These mice were vaccinated at 0 and 4 weeks. And then when we measured the HA-specific IgG using an ELISA at 4 and 6 weeks. So on the y-axis, we have our HA-IgG. And what we can see here is that we actually get a pretty good immune response from the pABOL, so just over 10 to the 4th nanograms per mil. But with our lipid nanoparticles, we see orders of magnitude higher for the antibody responses. So our best performing LNP was just over 10 to the 7th nanograms per mil. So we did a challenge study with these mice to see how well these antibodies were actually protecting them from influenza challenge. And in order to monitor this, we looked at the body weight over time. So this is a really good proxy in mice to see how they're responding to the infection. So you can see with our naive mice that by day 5, they had all met the endpoint for this study. Whereas for our pABOL group, you see they do lose some weight, but all of them survived and were protected in the end, but you do see some weight loss here. Whereas for our LNPs that had the really high antibody responses, that we didn't see any weight loss. So they were completely protected from the infection. So this is really interesting because even though we're seeing such high protein expression from the pABOL, we're seeing that the LNPs confer just on much higher immunogenicity than our polyplexes. So one of the things we really wanted to look into was, can we really push this to a lower dose using this top performing LNP formulation? So we looked at a huge range of dosing starting at 1 micrograms and going all the way down to 0.1 nanograms of saRNA formulated in this lead LNP. We compared this to 1 microgram of pABOL and then a naive group. And one of the things we were really interested in doing was actually looking at so most of these were intramuscular administration, which is what we normally use for vaccines. But we also wanted to look at intranasal administration just to see if we see any antibody responses with this instead. And so in order to do this, in this case, we were using SARS-CoV-2 as a model antigen, and we looked at both the systemic and the mucosal IgG that we are seeing. So on the left is the systemic and on the right is the mucosal. So I'll walk you through this again. So you can see, again, a really nice dose response curve with our saRNA LNPs. So our highest dose is just over 10 to the 5th nanograms per mil. And actually, we're even seeing antibody responses at our lowest dose of 0.1 nanograms, although you can see there's a lot of variability here, which is typical of what we see when that dose is kind of not starting to work anymore. So similarly, we see an okay response with our pABOL, but this reflects what we saw earlier, which is that it's much higher with the LNP than our pABOL formulation. And what was really interesting is that we actually see a pretty good immune response antibody-wise with the intranasal administration. So just over 10 to the 4th nanograms per mil. So it's about an order of magnitude lower than the same dose administered intramuscularly, but we do actually see a pretty good systemic immune response with an intranasal administration. When we look at the mucosal site, again, we see it's lower than we saw systemically, but very exact same trend. So we see a high antibody response with our highest dose. And again, at our lowest dose, it's really starting to fall off there. What's really interesting is that with our intranasal administration, it followed the exact same trend. So we thought that maybe administering it intranasally would result in a higher intranasal response, but that's not what we saw. It's basically exactly reflective of the systemic immune response. So to really dig into this is some of the work that we've done and are continuing to look into, just to look at the reactogenicity and really what is the difference in immune response and how cells are sensing an LNP versus our polymer. And so in order to do this, we did a Luminex assay and we looked at 25 different cytokines just 4 hours after we administered these particles. So what we saw, and I'll walk you through the groups, we looked at 1 microgram of lipid nanoparticles administered IM, pABOL administered IM, the LNPs administered intranasally compared to a naive group. So we didn't see any detectable levels for some common cytokines like GM-CSF, interleukin-1 beta, or IL-2, 13, or 18, but we did see some overall trends, and I'll walk you through those now. So you can see for the interferon gamma, it's maybe a little elevated, but not significantly so compared to the naive. Same with IL-12. All of the groups had higher IL-4 than our naive group. And the main difference that I think is really interesting, and we're starting to dig into more now, is actually IL-6. So you can see with our 1 microgram of LNPs administered intramuscularly that we had an elevated level of IL-6, whereas for any of our other groups, they were exact same as the naive. So there does seem to be some difference in what the LNPs are triggering when administered intramuscularly, which we suspect is actually adding to the immune response compared to pABOL. So this is really interesting, actually, because in a way, it's kind of nice to know that this pABOL polymer is more immune silent compared to the LNPs, because it means that you could potentially add in some adjuvant effect or have a little bit more modular control over that. So that's ongoing work in my lab now. The next thing I wanted to transition to is really thinking about LNPs and where we've come from and where we're going. And my view on this is that there's still a lot to understand and explore in this design space. And I think this timeline sums it up really well. So this was from a review that was published 2 years ago now, but it kind of it goes over the history of mRNA development and lipid nanoparticles. And of course, many people on this webinar will be familiar with the development of lipid nanoparticles. Many of the work was done in Peter Cullis's lab here at UBC. But what I wanted to highlight here was the mRNA. It's kind of been a convergent evolution of the mRNA and LNPs, which I think really means we need to think about kind of how we design these things and using this rational design approach. So mRNA has a really interesting history, right? So, in 1990 was the first time that somebody used mRNA in a mouse to look at protein translation, and then, developed over the course of many years with many people working on this to have the first clinical trial of mRNA in 2009, the first in human test of a personalized mRNA cancer vaccine in 2017. And LNPs really came from a different place, right? So they really started with the development of liposomes, and many of these were used for small molecule cancer drugs before then being used for an siRNA drug Onpattro, which was developed in 2018. So they really came from different places, and only recently have LNPs been used for the delivery of mRNA. And really interesting talking to Peter and others in the field is that the LNPs that we use for mRNA now really have kind of evolved from the systems that were really optimized for siRNA delivery to the liver. And they kind of just happen to work for mRNA. And so I think there's still a lot that we can learn in this space. So one of the things we wanted to do to pursue this is really look at different formulations for delivering mRNA and saRNA. So going back to what I talked about at the beginning, they have a lot of structural similarities, but also saRNA is much larger than mRNA. So we were wondering if we can actually really use the same formulations for delivering both of these types of RNA. So in order to do this, we made a number of formulations. So I'll walk you through the different lipids that we use. So in this study, we used 3 different ionizable lipids. So MC3, which is in the approved Onpattro formulation, ALCO315, which is the ionizable lipid in the Pfizer and BioNTech vaccine developed by CQAS, and SM102, which is in the Moderna vaccine. We used 2 different phospholipids, so DOPE and DSPC, a PEG lipid and cholesterol, as well as 2 different types of mRNA, so mRNA and saRNA. So the questions we asked in the study were, are the optimal parameters for saRNA also optimal for mRNA? Can we use the same LNPs for both types of RNA? But also, can we modulate the cellular activation by changing the LNP composition? So this was really building off of what we've seen with the pABOL and LNP studies, and just wondering if we could modulate this by changing the different lipids that we're using. So in order to do this, we did a design of experiments optimization. So this is a classic engineering approach to look at many different factors and how this affects the output formulation. So I'll walk you through kind of our inputs and outputs for this experiment. So our inputs, we looked at many different things, including the N to P ratio, the phospholipid type and content, the ionizable lipid type with the pKa's there for your reference and content, different amounts of PEG, but we also looked at some of the actual formulation parameters. So the process parameters, so the total flow rate, the temperature during formulation, the pH during formulation, and of course the type of RNA. For the experimental outputs, we looked at kind of some of the typical characterizations, so the size, the PDI, the encapsulation efficiency, and the charge. But we also wanted to look at the percentage of filled particles, so how many particles actually have RNA in them, as well as the percent of full RNA transcripts. So how much of the RNA that we put in is full length at the end. So on the right, we have this heat map of basically the significant factors for all the different outputs here. And so this was recently published in Molecular Pharmaceutics, so feel free to go read it if you'd like to look into it in more detail. But we've also, one of the things that really stood out to me in this is looking at the RNA type and how much that impacts the RNA integrity. So this was kind of a huge red flag for us, which is basically telling us that the longer the RNA, the harder it is to encapsulate. So I'll walk you through our optimal formulations here. This is a typical output from a DOE. And so we have kind of 3 different optimized formulations here. So the first one is to minimize cellular activation. The second one is to maximize cellular activation. And the third is to optimize our critical quality attributes. So this is the size, charge, PDI, et cetera. And over here, what's really cool about these different DOEs is we can make this 3D design space. So really get a sense of how, if we change the ionizable lipid content and the pH, how does that change the protein expression that we see, but also the amount of IL-6 that's released. And one thing I wanted to really highlight here too is that when we looked at the protein expression from all our different formulations with these 3 different ionizable lipids, what you'll notice is that both the ALC-0315 and SM-102, so these are both the ionizable lipids used in the Pfizer, BioNTech, and Moderna vaccines. They had over an order of magnitude higher protein expression on average than MC3. So I think this really kind of governs what we consider the state of the art in the field, which was MC3 up until recently. But I think now we need to kind of consider these different ionizable lipids as our controls or standards. So what you'll notice about our different optimal formulations is actually they have very different inputs for achieving these different goals. So we want to minimize cellular activation. We use ALC-0315 and a lower pH in our formulation. Whereas if we want to maximize cellular activation or the amount of IL-6 that's released, we use SM-102 and a slightly higher pH. So we went back and validated these different parameters, so different optima. So again, it's minimizing and maximizing cellular activation and then meeting our CQAs. And this was our predicted value, and then we verified it and validated it for both mRNA and saRNA. So there's some things that are pretty spot on here. So you'll notice for the size, it's pretty close to what we predicted for both the mRNA and saRNA. For the RNA integrity, or the percent of full length of transcripts, you'll see that the predicted was pretty spot on for the saRNA, but the mRNA was actually much higher. So what's really striking about this is for any of these optima, you see that we only have about 50% full length saRNA. So this means, 50% of the saRNA going in is just causing side effects. It's not resulting in any effect. And so this is obviously something that we're working hard to optimize now. One of the things I want to point out that was really useful about this modeling is that you can see for our predicted IL-6 release, this was actually pretty much the same for mRNA and saRNA. And we're able to increase this by almost twice as much just by changing the lipid composition. So I think this really goes to show that we are able to modulate how much this cellular activation that we're seeing. So to sum this up, we really see that changing the formulation parameters can directly impact the cellular activation. So in this case, the IL-6 secretion. We do see that the optimal formulations for saRNA are also suitable for mRNA. So that's an okay assumption to make, at least with these formulations. We see that the size greatly affects the integrity after encapsulation. So this is important to keep in mind, not just for saRNA, but for other types of long RNA, such as CRISPR, which is much bigger than a typical mRNA. And we see that our saRNA, as expected, is about 100 to 1,000 times higher protein expression than mRNA. So conclusion for this section, and then I just have one more vignette talking about the saRNA design, is that for our formulation strategies, our polyplexes result in about 100 times higher protein expression, whereas the LNPs are about 100 times higher immunogenicity in the context of a vaccine. The route of administration makes a big impact. So the IM administration always results in about 10 times higher antibody responses than the intranasal administration. And when we are optimizing mRNA and saRNA LNPs, we see that tuning the LNP formulations can directly impact the cellular activation, and that these large RNAs are really susceptible to degradation during encapsulation. So the last thing I wanted to do is really think about kind of the molecular design of RNA. So I think, we think a lot about these LNPs and increasing the transfection efficiency that we see with them. But for us, the RNA design and the LNP or nanoparticle design really go hand in hand. And so we also thought about how is this RNA sensed intracellularly, and how can we actually help to modulate this by using the RNA design itself. So coming back to, these typical ways that RNA is sensed, so saRNA is sensed in similar ways to mRNA, so these endosomal sensing and cytosolic sensing pathways. So one of the things that we considered in this study, which I don't think I really need to convince anybody of, is that humans are not mice, ferrets, or monkeys. But I wanted to walk you through some of the data that I think really shows this and why it's important to choose the model that we're optimizing in for these different delivery systems. So this was a study that was published in 2017 from Moderna, and I'm always surprised that the lawyers let this out the door, but I think it's a really important contribution to the field. So in this case, they made a flu vaccine, and the correlative protection for a flu vaccine is an HAI titer, a hemoglobin inhibition titer. And the higher it is, the better the vaccine works. So they did this first study in mice, and they see a really good immune response. The HAI titer is about 1,000. Just FYI, the correlative protection for humans is an HAI titer of 40 or more. So they then went on to do this in ferrets. And again, they see a really high HAI titer of almost 10,000. So they moved this into non-human primates, and they saw an HAI titer, again, very high, almost 10,000. So they moved this into a human clinical trial and looked at the HAI titer, and you can see that they're much lower than any of the preclinical animal models. So the average was below 100. So this is really important to consider. We've seen these differences over and over again between any preclinical animal model and humans, especially for RNA, which sense RNA in very different ways. So one of the things we wanted to do was see if we can modulate this innate immune response by using interferon inhibiting proteins, or IIPs, that are derived from viruses. So there's many viruses that have evolved to have these proteins to modulate the interferon response. So we wondered if incorporating these directly into our saRNA would be able to kind of tamp down the reactogenicity that we see. So in order to do this, we have our wild type saRNA, or replicon, at the top. And then we designed a library of interferon inhibiting proteins and put them directly into our saRNA right after our gene of interest using a T2A cleavage site. So the first thing we wanted to do was see if the IIPs enhance protein expression in vitro in human cells. And we used a number of different cells, and I'll walk you through why. So on the left, we have HEK cells, and then HeLa cells, and MRC5 cells. So HEK cells are known to not have an intact interferon response, and so we wouldn't really expect to see a difference here. Our black is our wild type, and then all of the different colors are our different IIPs that we tested from our library. And we did 2 batches of RNA just to make sure there wasn't variation between the two. And you'll see the HEK, there really was no difference in any of the expression that we saw. For the HeLa and MRC5 cells, both of these cell lines are known to have a more intact interferon response, so we would expect to see differences. And there were really 2 standouts in both of these cell types. So one was a protein from pID5, and the other is a protein actually from MERS called ORF4A. So we then went on to look at some different studies. We did study this in mice, and for any of you, those of you who have worked with RNA, you'll have probably seen the similar kind of parabolic relationship between the dose of RNA and the expression that you see. So with any type of RNA, there gets to a point where there's too much RNA, and you stop seeing a beneficial effect of increasing the dose. So in order to do this, we looked at our wild type fluke and our IIP. And we looked at 3 different doses, so 0.2, 2, and 20 micrograms of both 7 and 10 days. And what you can very visibly see here with our wild type is that when we go from 0.2 to 2, we do see an increase in the protein expression. But when we increase the dose to 20 micrograms, we actually see very little expression because it's just too much RNA. With our IIP saRNA, we see very similar levels at the 0.2 dose. When we increase to 2 micrograms, the protein expression is slightly higher. And what's really important, I think, is actually when we increase to 20 micrograms, we're still seeing some protein expression. So this fairly partially abates or partly rescues that dose dependence that we see in vivo in mice at least. But again, mice aren't a great model for any RNA to test it. So one of the things we did and continue to do in my lab is look at different RNA formulations in human skin explants. So these we get from local plastic surgeons who would regularly discard the tissue, but it's actually a great tool for us to, because we can look at how much of the protein is being expressed in a human a native architecture with a native innate immune response. So in this case, we're using GFP as a reporter protein and looking at the percentage of GFP positive cells. So again, we have our wild type saRNA and then our 2 IIPs. And again, you can see here, we use the exact same doses that we did in mice. And you can see that when we go from 0.2 to 2 with the wild type, we see a slight increase in the number of GFP positive cells. But when we increase it to 20 micrograms, it goes down. So this is a very typical response. We've seen it in basically every single system I've ever tested this in. And so when we looked at our IIPs, similar, when we go from 0.2 to 2, we do see an increase. And what was remarkable actually is that when we go from 2 to 20, we see an increase with these IIPs. So not only are we not going down, but it's actually going up, which I think speaks to really how important this innate immune response is in human tissues. So we went on to test this in a rabbit vaccine model, just because we know we didn't see much of a difference in mice, and we wouldn't see a big difference, at least in the strains of mice we were using. So in order to do this, we used rabies as a model antigen, and these rabbits got injections at 0 and 4 weeks. And then we looked at both the antibody response and the neutralization at 0, 4, and 6 weeks. In this case, we were using the wild type and the MERS-ORF4A protein. You can see from the antibody data that we do see a slight increase after 4 weeks, but we do see about an order of magnitude higher after 6 weeks with our IIP compared to our wild type. And this is directly reflected in the neutralization assay as well. So it's much higher, about an order of magnitude higher than our wild type saRNA. So not only does it increase the protein expression that we're seeing, but it also does result in an increased antibody titer in a more interferon competent preclinical model. So in conclusion, with our saRNA, we see that it results in about 10 times higher protein expression compared to mRNA in vivo and in vitro. And for the interferon inhibiting proteins, we identified 2 that abate this nonlinear dose response in human skin explants. And we also see that they result in a higher vaccine immunogenicity in a preclinical rabbit model. So the last thing I wanted to finish on is just thinking about kind of what we're trying to do in the context of NMIN, as well as where the field is going. So this is actually not my slide. I took this from BioNTech's website, but I think it does a really good job of portraying kind of where we are and where we're going. So, today we have these 2 approved mRNA vaccines, things BioNTech is already actively working on. So mRNA vaccines for other infectious diseases, cancer vaccines, CAR T-cell amplifying RNA vaccines, as well as immunotherapies, but really thinking in the future about autoimmune diseases, allergy, inflammation, regenerative medicine. And so these formulations and thinking about the immunogenicity is just going to get more and more important as we use these for applications beyond vaccines. So with that, I'd like to acknowledge all the people that contributed to these studies. So my lab here at UBC, in particular, Hannah and Kay, who were my first students who published the molecular pharmaceutics paper, as well as our collaborators at Imperial, Precision, and Acutis, and our funding sources. And with that, I will take any questions.
Terry Allen asks, since mRNA and siRNA have differences in PK, response and duration of action, etc., have you looked at combining the 2 either in the same formulation or in 2 different formulations administered together?
Great question, Terry. We actually just did that study a few weeks ago and are analyzing the ELISA results from it today. So yeah, I think it hasn't been done before or published before from what I've seen, but I think it's actually really interesting to look at, okay, if you get that initial burst from the mRNA, but then the duration from the saRNA, do you see a better immune response? So something we're actively doing, and maybe I'll report it in my next NMIN presentation.
Yeah, great talk, by the way, and I really enjoyed it.
Thank you.
And I just wonder, if combining them not only would smooth out the response level and duration, but also would allow you to use lower doses of either one and maybe reduce side effects.
Yeah, great question. Yeah, something we're actively looking into. I think there's a lot of potential there.
Great, thanks, Terry and Anna as well. Afsana has a question.
Hi, Anna, great talk, and I really enjoyed it. So I had two questions. One was about the polymer that you have used, and I was wondering why, despite having higher protein expression, you didn't have as much immune response compared to the LNPs. And then now that Terry asked about combination, I was wondering if you are thinking about combining the polymer with the lipid and maybe get the benefit of both. Yeah.
Yeah, good question. Yeah, interesting, all these combinations that are possible, right? So yeah, it's a really interesting thing seeing the higher protein expression with the polymer, but the higher immune response with the LNPs. So we're actively looking into this now, and I have kind of two theories about why this might be. So if you remember, one of the main differences between the particles is that the polyplexes are positively charged, whereas the LNPs are neutral, right? So I think that may affect their biodistribution, and I suspect that the LNPs may be more efficient at draining to the lymph node. We know that neutral particles typically are better at that than a charged particle. And so even though we're seeing higher protein expression in the muscle, it may be more important for a vaccine to actually have expression in the lymph node. This is kind of one of the challenges in RNA vaccines right now, is we actually don't know what the target cells are. So whether that's APCs in the muscle or actually draining directly to the lymph node. So that's something we're looking at now. The other component of it, I think, is really important, is actually, or kind of maybe underappreciated in the field, is actually just how important the adjuvant effect of the LNPs are. So that was another thing that we saw with the cytokines, right? So for sure, the LNPs are triggering that IL-6, which I think is really important for that immune response. And with the pABOL, something we're looking at now is can we add back in an adjuvant response? I think it may actually be beneficial to be able to add something in to directly, there's so much known about different adjuvants and how you can kind of direct that in different ways depending on the immune response you want. So now we're looking at, okay, can we actually add in more immunogenicity for the pABOL and kind of harness that high protein expression we're seeing, but make use of that. But yeah, it's a really interesting point, actually, looking at, we've never looked at combining the pABOL and the LNPs, but if you think about like how viral infections work, it may be beneficial to have both of them, to have like a high antigen expression in the muscle and the LNPs potentially draining to the lymph node. So I think we have a lot to learn, but yeah, it's like very good questions.
I have one more question. If there's, you looked at IL-6 as a measure of cellular immune response, is that correct? So I wonder, like IL-6 is always considered to be a double-edged sword, and like, have you thought about looking at IL-12 or like why IL-6?
Yeah, IL-6 was just one of the ones out of the panel that we saw differences in. And so when we were then like studying LNPs with the different compositions, we just chose that because we knew we saw differences in that. But you're right, it would be really interesting to do kind of a more comprehensive study and look at all the different cytokines and kind of what's modulated in response. I think one of the challenges in the field right now is we don't really know how to design different adjuvant effects with these LNPs. And I think it's even just starting to be appreciated that the LNPs actually have a really profound adjuvant effect. Like there's kind of just a handful of papers out there right now. So yeah, it's a really good point. I'd be interested to look at like a more comprehensive set of cytokines.
Yeah, thanks a lot.
Thanks very much. There's a question from Javed Sadeghi. Javed, do you care to ask your question? Okay, I'll ask for Javed. Very interesting talk. You almost answered my last question in your last slides, but I was wondering how saRNA vaccine can help with the viruses that we could not have had vaccine for them, such as HIV, as they also use different replication strategies.
Yeah, that's a great question. One of the things, kind of like the caveats, I always say is like, RNA vaccines are a tool, right? So I think some of the challenges, so I actually did part of my PhD was like really focusing on HIV prevention. And so I learned a lot about that field. But I think the challenge with HIV vaccines right now isn't that we don't have a good platform. It's just we don't know what that optimal antigen is to induce broadly neutralizing antibodies. So, there's lots of work being done with mRNA vaccine now. Moderna has an HIV vaccine that's going into clinical, or I think it already is in clinical trials. I'm just not sure that we've had kind of the advances of the antigen design to actually make these kind of different vaccines that we don't yet have vaccines for work yet. So one of the things that I think is kind of underappreciated in kind of the COVID vaccine space is one of the reasons that we had vaccines so quickly was because all of the work that Jason McClellan and the NIH had done on designing that spike antigen to make it more immunogenic for a vaccine. So if we hadn't had that, I think it's very unlikely that we would have had any approved vaccines so quickly. But I think that's still kind of the missing link for HIV is, if there was an antigen that worked really well for inducing immune responses, I think you could plug that into an RNA platform and use that, but we just don't have that antigen yet. So I think that's a part of it that's also really important is kind of that structural biology antigen design. So RNA is not like the solution to all of our vaccine problems right now, but I think it's a tool going forwards.
Hello, thank you. It was great talk. I learned so much. But I was kind of curious, when you make a vaccine, you want immunogenicity. And when you look beyond vaccines, you don't want immunogenicity for a lot of other kind of therapeutics, right? So would you be kind of optimizing in the opposite direction for those sort of applications? Or like, how would you change your optimization if you were looking at kind of that last slide where you were saying, beyond vaccines, like what for the other applications, how would you be optimizing your LNPs or your PNPs to lower your immunogenicity?
Yeah, great question. So I think you're spot on. For any like therapeutic application, you actually don't want any immunogenicity. And actually for the RNA vaccines, like it's a balance, right? You just want the right amount to have an immune response, but not too many side effects. So yeah, there's it's an interesting thing. I think that's actually why there's kind of growing interest in the polymer space, because like fundamentally, LNPs will always have an aspect of immunogenicity, right? Just because of the way lipids are sensed within cells, even these like synthetic ionizable lipids that don't occur naturally in nature. So, there's many tools you can use to do this. One of the things we've started looking into recently is actually including different like small molecules in these formulations, which are known to tamp down that immunogenicity. So I think, yeah, it's strategies like that on both the RNA and the nanoparticle side, but I think it's super important for therapeutics.
Thank you so much.
Thanks for the question.
Thanks, Liza. Maybe I'll just squeeze in one last question. I see we have about a minute left. So when you load the messenger RNA too high, you have a side effect, and that drops the titer response. Can you tell us what's going on there on the upper end?
Yeah, so it's basically the same mechanism that has evolved from our bodies recognizing viruses, right? So when you detect foreign RNA, if there's too much of it there, it just shuts down all the translation in that host cell, because it knows that the virus is basically trying to replicate more virus. And so that's our immune response against it. So it's the exact same thing when we deliver a messenger RNA. If there's too much there, it's detected as a foreign RNA and just completely shuts down the protein translation.
Great, thanks very much.